Okay, so step number 2 on page 50 says place 4 tubes in a test tube rack and label them 1, 2, 3, 4. I did that. And then fill each tube with 10 milliliters of your hydrogen peroxide. This is how I do that. This is my hydrogen peroxide. Has a little plunger. Pick it up, I push it down. Gives me 10. Kind of cool, huh? I know, it's exciting. One of the things it's good to do this for is that then you can check, do all your tubes have about the same level? This one's a little bit lower than those other ones, so I'm going to see if I can give it just an extra little smidge here. Not quite. A little bit more. Sometimes you want to have all the same, you want to have as close to the same amount of substrate in all of your tubes so that you don't have to think like one tube has more than the other and that could throw your results off. Then it says you're going to, if you go to step 3, pour the 10 milliliters into an Nalgene bottle. I'm going to use this flask instead. I'm going to pour it in here. Put the flask on the table. And then it says using the labeled pipette, you drop some of the enzyme suspension. Okay. This, remember, was my enzyme I mixed up, and you can see it's getting all, see it's all bubbly. Tells me my yeast has awoken. I have a little, it's in a little flask that says enzyme. It says, it's when it says resuspend each time, what they want you to do is just make sure you mix it. Then they want you to put 1, 2, 3, 4, 5 drops in, right? And you can already see if you look closely, it's getting kind of bubbly. You want to put the stopper in here and then go over to the computer and click. I'm going to switch cameras here quick so you can see this. Oop. I'm going to switch cameras and show you how to do the collection. So stop it. Do exactly what I did. Pull up a thing and do a thing and clear all data. Now you're going to have to edit. Once you have your whole little thing set up, you click the collect button here and it's going to collect this data, you'll see, in this little table here every few seconds, and it's going to do it for 5 minutes. And once 5 minutes is up, I'll show you what it looks like. So this is the little setup I did. I don't know how well you can see this, but there's tiny little bubbles in the bottom of the flask. That is because the enzyme, right, is taking in the hydrogen peroxide and splitting it apart, and the little bubbles are the oxygen it's releasing, which that is what the magic wand oxygen meter is measuring the release of. And then all of that gets measured on the computer over here. So I'm back looking at this again, and you can see this has stopped measuring because it's been 5 minutes, and it's plotted this beautiful almost straight red line from all this data over here on this little table. And the table, you can scroll, this is the very beginning. So at the beginning, the amount of oxygen gas in this flask that was measured by the magic wand was about 13%. And you can see it stays 13% for a pretty long time. But eventually that number goes up to 14%, which isn't a huge change, but By the end, it's 14.8, which is almost 15%. So this is a really super gradual rise in oxygen concentration over time. And that was with the 5 drops of enzyme. Now we're on to the second time through this experiment. We're on step 6 of page 51, which says repeat steps 3 to 5 using 10 drops, 20 drops, 0 drops. So for the second one, I'm going to put in 10 drops of enzyme. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I'm going to go swirly whirly, and I'm going to put in my oxygen sensor again. And then over here on the computer, I'm going to press go again. So you'll see for the second run, it's going to plot that data in blue. First run was red. And already you can see this blue line is a little bit above that red line. Okay, so here's the results from the run. The blue run is the run with 10 drops of enzyme. You can see on this one, it started at 14%. And it went down up to, let's see, not a huge amount. Oh my goodness, this is so hard to scroll. 16.8. And clearly it's gone up more steadily than the red line did. Okay, now I'm going to do 10 milliliters of hydrogen peroxide with 20 drops of enzyme. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So you can see, if you kind of carefully do the math, we started at, what was it, 5, then 10, then 20. So we've doubled it every time. I thought I'd take a short video too of this. This is the hydrogen peroxide with 20 drops, and you can see the solution bubbling away. Many, many more bubbles than it had, say, the first time. Okay, here's the results for the third run with 20 drops. You can see right now the oxygen percent's at about 21. But for this run, again, it started at 14. It's where we've started almost every time. And we made it up to about 20. Almost 21%. And this is the green line now. Green line is the 20 drops of enzyme. Clearly a lot more oxygen produced than at 10 or 5 drops. So the last round for this set of the experiment is just hydrogen peroxide and no enzyme. So we're going to put the hydrogen peroxide in. You can see, kind of boring, no bubbles. You tell me, what do you think is going to happen? Okay, so the last run is done. It is a little hard to see, but it's this orange line right underneath a red line. And if you look over here at the percent oxygen, you'll see we started at 14%, and basically, if I can get this to scroll down, we ended at 14%, so basically no change at all, which is not surprising because we didn't add any enzyme to it. So now I am on step 7 of 51, and it says using the tangent button at the top of your Logger Pro. So that's this button. If I hover, if I can get it to show up. If I hover over it, it says tangent. So click that, and what it's going to do is it puts these little tiny black cursors along the lines, and you can drag them out. And what it does is it calculates the slope, which is kind of the calculation of the rate of O2 production at different time intervals. what you want to do now is go to table 2 on page 51, and it says at 0.5 minutes, what was the rate of production for 5 drops, 10 drops, 20 drops? So I'm going to go to 0.5 minutes right here, and there's this teeny tiny little box up here that tells me my rate at 5 minutes, or I'm sorry, 0.5 minutes for 5 drops is 0.206%. For 10 drops, it's the blue line at 0.433, and then for the green, it is 1.224. For the 0, it is 0.0, essentially 2. And I'm going to do this again at 1 minute. I'm going to write down the numbers. I'm going to do it at 1.5, 2, and I will take a picture and show that to you on the lab.